on a huge talent for school and for science in particular. For families like his at that time, of limited means, there was one obvious route to a further study, and that was to join a monastery. And so in 1843, Mendel entered the Augustinian monastery in Brunn. Now, I say monastery, and you might think tonsured monks hiding themselves away in little cells, spending all day in prayer. Not at all with the Augustinians in Brunn. They were a worldly bunch and a playful bunch, as you see here in a group photograph taken in the 1860s. There's Mendel there with the letter M. The Augustinians were very and are very worldly. Their mission was to help, to educate in the first instance, but also to be engaged, not least scientifically. So the Augustinian monastery in Brunn was a site of a sort of permanent scientific philosophical seminar. It was an exceptionally lively place that Mendel had joined, and he became a teacher there. And through the offices of the monastery, he ended up studying.
Ma,
Oh, uh, through the offices of the monastery, he ended up studying science for a science degree at the University of Vienna in the early 1850s. So again, we have to get away from this idea of an isolated figure, isolated, culturally isolated scientifically. On the contrary, at the University of Vienna in the early 1850s, Mendel got one of the best scientific educations available on the planet. One of his teachers was Christian Doppler of the Doppler effect. He studied physics with Doppler, so he got an exceptionally good scientific training at the University of Vienna, returned to Brunn, uh, returned to his teaching duties, was a very successful teacher, but he also, in the mid 1850s, began the set of experiments for which he became, much later, remembered. Experiments done with varieties of the garden pea, Pisum sativum. I'll be saying more about those in a moment. He wrapped up his experiments, gave lectures on them in 1865, published the results in the Brunn Proceedings of Natural Science in 1866. At that point, he got promoted to abbot of the monastery. And though this was very good for Mendel, it wasn't so good for his science, and his scientific activities really dwindled from that point onward. He was a successful abbot, and he died in 1884 with a great deal of local esteem, but not widely known beyond that. Things changed for Mendel after his death. In the year 1900, famously, as every biology textbook reports, three European botanists rediscovered Mendel's results, rediscovered his explanations for those results, and suddenly the monk that almost no one had heard of outside of Brunn became a talking point. And over the next 10 years, he went from being obscure to being one of the Olympians of science, the founder of a new science, genetics. Genetics being the science of inheritance founded in the image of the experiments Mendel did in the monastery garden with his peas. Now, for those of you who, again, haven't maybe thought about this in a little while, I thought it'd be useful to go over the basic elements of those experiments. So, as every textbook will tell you, Mendel spent years purifying his starting materials. So he's working with the garden pea, Pisum sativum, and he winnows the different varieties he's working with to varieties that show these either or traits. So in this case, we're looking at seed color. Either the seed is yellow or it's green. And Mendel spent years purifying his starting breeds, making sure that his yellow-seeded varieties only ever produced yellow-seeded progeny, that they were, in the jargon, true breeding. And likewise, that his green-seeded varieties only ever produced green-seeded progeny. Only at that point did he begin the crossing experiments that were his goal. And on crossing a yellow seeded variety and a green seeded variety, he found that in the next generation, all of the peas were yellow. Now, when he let those peas self-fertilize, in the following generation, green came back. And it came back in a remarkably regular ratio of 3 yellow peas to 1 green pea. So this is remarkable enough in itself, right? A new empirical pattern. But Mendel is really just in the foothills here of his experiment because he goes on from there to explain this pattern very elegantly. Mendel says, let us suppose that in the yellow seeded varieties, there is nothing but yellow making stuff when it comes to color of the seed of the pea. And likewise, let us suppose that in the green seeded varieties, there is nothing but green making stuff. So when those varieties produce gametes, produce pollen and eggs, there is nothing but in the yellow seeded pea, yellow making stuff, let's call that big A. And in the green seeded variety, green making stuff, call that little a. Now, what happens when crossing is undertaken is that the yellow making material, big A, and the green making material, little a, are united, right, in the hybrid. But of course, all we see is yellowness. What, how do we characterize that? Well, Mendel says, let's call yellowness dominant. All that means for Mendel is that it is the character that's visible in that first hybrid generation. And greenness, let's call recessive, which is just to say it recedes, right, from visibility in that first hybrid generation, but it's going to come back again. And now the magic happens in Mendel's explanations of what he's observed. He says, let us suppose that when those hybrid peas themselves produce gametes, produce pollen and egg cells, the gametes are not themselves hybrid, right? They're not greeny yellow. They're not big A, little a meshed together. On the contrary, the gametes are pure for either yellow making stuff or for green making stuff, for the big A or the little a, right? So the gametes are not themselves hybrid. And let's suppose further, Mendel says, that it's a matter of chance, right? Purely random, which egg cell meets with which pollen cell. With relatively simple mathematics, Mendel's able to show that there are just 4 possibilities. Let's walk through one by one. One possibility is that yellow making stuff meets yellow making stuff, right? Big A meets big A. In that case, the pea seed is going to be yellow. Another possibility is that big A meets little a, right? Yellow making stuff meets green making stuff. Yellow beats green, right? Yellow is dominant, and that pea is going to be yellow. A third possibility is just a version of what I just gave you, right? Green making stuff meets yellow making stuff. Again, yellow beats green. That pea is going to be yellow. Only in the one case where green making stuff meets green making stuff, where little a meets little a, will the pea be green. So we get explains now that 3-to-1 ratio of yellow peas to green peas in the second hybrid generation. And in my view, if you like science at all, and not everybody does, but if you like science at all, you feel a little pop of pleasure at that moment. How clever, one thinks. How clever of nature to work so elegantly. How clever of Gregor Mendel to have figured that out. And for those of you who are familiar with the paper, familiar with genetics textbooks, you know that Mendel goes on to elaborate all of this with enormous ingenuity, but these are the basics. Now, one doesn't have to study formally in school to learn about all of this. Mendel's results have percolated throughout the wider culture. It's the subject of gags. This is from a horrible science book that my kids had. We see here the inevitable pea gags. There's Gregor Mendel in the refectory at the monastery telling the other brothers that there's no peas for the wicked. Here's a question for you. What did Gregor Mendel say when he founded genetics? Think about it. I can't hear your answers, but some of you, I hope, will have guessed that he said whoopee. Right. So again, we have here a very appealing figure, the subject of much amusement, but also the subject of myth making. And I'm here tonight to help correct some of the myths, and I'm going to concentrate on two of them in particular. The first myth that I'm going to confront is the claim that Gregor Mendel discovered the gene, considered as the atom of heredity. The second myth, related to the first, is that enlightened understanding of biological inheritance uh, in the history of science, as in the science classroom, starts with Mendel. Right? That's the beginning. That's the received wisdom on Mendel, the myths that I want tonight to disabuse you of. That's my aim. And I'll come back at the end to say a little bit about where that leaves Gregor Mendel in our affections and our esteem. I should say that I'll be drawing throughout tonight on material that you can find set out in greater detail in the book that Josh mentioned, Disputed Inheritance, The Battle Over Mendel and the Future of Biology. And you'll see on the cover of the book is an image of peas, but they're not quite the peas that Gregor Mendel prepares us to expect. I'll be coming back to that in a little bit. So first, the notion that Mendel discovered the gene, the atom of heredity. In the view of many historians, and here I endorse their work, the key to getting straight about what Mendel thought he was doing, and so overcoming this myth, is in the first instance to concentrate on the title of Mendel's paper, the title of the lectures he gave in 1865, the title of the published paper in 1866. We see it here in the original German, Versuche über Pflanzenhybriden, Experiments on Plant Hybrids. For the historian of science, the key to unlocking the historical Mendel, as opposed to the ahistorical Mendel, the Mendel of our textbooks, the Mendel of myth, is to take really seriously that notion that it's about not heredity, which is a word that barely comes up in the paper. It comes up once in passing, but hybrids. So what is it to say that Mendel's experiments were in the first instance aimed at illuminating not heredity, but hybrids? Well, Mendel tells us that in his view, there are two great classes, two great kind of categories of hybrids. One he calls the constant hybrids. They're of the following type, right? You've got two plants with interesting characters. Let's call that species A and species B. You cross them together and you get an offspring with this new exciting character C, right? And if you're a breeder, that's potentially going to be commercially important for you. So what you hope is that having produced a plant with this new exciting character C, you cross your fingers and hope that when you let that plant self-fertilize, the new character is going to carry on, to be perpetuated. So you cross your fingers, self-fertilize, and you're in luck. It works. The hybrid character stays constant, generation after generation after generation. Mendel says this is not what my paper is about. My paper is not about the constant hybrids. Rather, my paper is about this other class of hybrids, where you cross the species with character A and the species with character B. You get a species with interesting character C. You cross your fingers, let the plant self-fertilize, and it doesn't work out. The old character comes back again. Mendel says, this is what my paper is about. What Mendel's trying to understand is whether there is a law that governs the fate of the hybrid character in these variable hybrids, right? Where the hybrid character doesn't stay constant. Is there a law that governs the fate of that hybrid character? And if there is a law, can he explain it? That's Mendel's aim, and he achieves the aim amazingly. Now, in my view, an interesting test of this interpretation of the paper is to have a look at Mendel's copy of Charles Darwin's On the Origin of Species, published in 1859, right? So Mendel's doing these experiments between the mid 1850s and the mid 1860s. Right in the middle of that, Charles Darwin publishes On the Origin of Species. And within a few years, there is a German translation. And we know that Mendel owned that translation, and indeed, his copy comes down to us today. And so you can examine Mendel's copy of the German translation of Darwin's Origin. Now, chapter one of The Origin of Species has some magnificent passages on inheritance. How many annotations do you think Mendel put on the passages on inheritance, on heredity in The Origin of Species? The answer is zero. Almost all of his annotations on the origin of species are in the chapter that most of us nowadays don't read, a chapter in the middle about hybrids. Hybrids is what Mendel was interested in. When he read The Origin of Species, he had hybrids in mind. And when he let the pen go to make notes, it was the bits about hybrids that got him stimulated. So I think that's, a pretty good test of this interpretation. So it's not about heredity. Let's come now to the other aspect of the myth, right? Atoms of heredity. The notion that Mendel conceived of his underlying factors, the A making, the green making stuff, the yellow making stuff, big A, little a, in the way that we think about genes as discrete entities, as atomic, right? Atom-like. Well, you can read the paper uh, on the supposition that Mendel thought of the underlying materials as atomic, but there's no need to do so. And indeed, everything in the paper is consistent with the thought that Mendel didn't think about the materials in that way. Indeed, that he thought about yellow making stuff and green making stuff as like oil and water, as immiscible fluids. Just to go back again through the basic experiment, right? When we get to the formation of the hybrid, right, where yellow making stuff and green making stuff come together in that hybrid, when you read what Mendel has to say about it, he has a vision of these as coming together as an unstable union, its contents under pressure. Right? They don't lose their identities. And when gametes form in that first hybrid, it's like the release of pressure. That's what's happening there, right? The oil and the water separate, the yellow making stuff and the green making stuff separate. So there's no license historically for reading the paper as putting forward an atomic conception of the underlying materials. On the contrary, there's no license for that at all. And everything Mendel says is consistent with his having, for example, this fluidic conception. So let me summarize this part of the talk by identifying some interim take-home messages. The first, to quote a former colleague of mine at Leeds, Bob Albee, put this as the title of a famous paper. Mendel was no Mendelian. This shouldn't really surprise us, right? Marx was no Marxian. Likewise, Mendel was no Mendelian. His paper wasn't about heredity, and his conception of his materials, the underlying material, wasn't atomic. And indeed, as I've emphasized, the reason he concentrated at the start on these either or uniform characters, right? Yellowness, greenness, roundedness, wrinkledness, and so on, caused by nothing other than associated cellular material, was because he was interested in tracking the fate of the hybrid characters, right? Mendel's choices are brilliant choices for someone with his questions. If what you're interested in is tracking the fate of the hybrid character, right? Figuring out whether they follow a law and then explaining that law, concentrating on these either or, these blockily binary characters, which are caused by nothing other than the cellular material within, is absolutely brilliant. But those same choices, construed as starting points for understanding inheritance, right? Hereditary characters have been and remain very problematic. And that now takes me to the second myth, that enlightened understanding of biological inheritance, right, in the long run of the history of science, as in the science classroom, rightly starts with Mendel. And here now I'm going to give you the larger photograph that the image on the cover of my book, Disputed Inheritance, draws from. If you have a look at the top row, we see in box number 1 peas that are really, really green, and in box number 6 peas that are really, really yellow. But in between, we see a spectrum, a color continuum, from deep green through to greeny yellow onto yellowy green, and then finally to yellowness. It appears, in other words, that if you're not committed in advance to describing peas using Mendel's binary categories, you won't necessarily see a binary reality. Now, this image comes from a paper published in 1902. So that's 2 years after the rediscovery of Mendel's paper. And it was published by this man, Walter Frank Raphael Weldon, WFR Weldon, Raphael to his friends, born in 1860. By the year 1900, he was professor of comparative anatomy at the University of Oxford. And from that year, 1900, Weldon took an interest in the buzz around Mendel's paper, but he rapidly grew skeptical. And in the early months of 1902, he published a critique of Mendel's paper, which included that photograph. Now, in doing so, he infuriated his former friend turned frenemy, turned now proper enemy, William Bateson. Weldon, as I said, was based at Oxford. Bateson was based at the other great ancient university in England, Cambridge. Bateson, too, took an interest right from the start, right from the spring of 1900, in Mendel's paper. But far from becoming skeptical, Bateson convinced himself that Mendel's paper changed everything, that Mendel's paper was revolutionary, revolutionary for biology, and because it was revolutionary for biology, it was going to be revolutionary for society. And he rapidly turned himself into the greatest champion of Mendel that there ever was. And the Mendel that we know from our textbooks, and indeed the choreography that we know in thinking about the way that Mendel's experiments works, are very much the work of Bateson and the brilliant group of students that he rapidly attracted around him in Cambridge to help push forward the new Mendelism, as it comes to be known, or from 1905, 1906, genetics, genetics being Bateson's coinage. So these two from 1902 went at it furiously because there was something very important at stake. How was the 20th century science of inheritance, surely going to be quantitative, surely going to be experimental, how was it going to be organized? Around what? Was it going to be organized around the blocky binary characters that feature in Mendel's experiments and the kinds of explanations that Mendel favored, right? Explanations in the first instance exclusively in terms of those underlying factors, or as we called them from 1909, genes? Or would there be something different? Weldon began moving towards the alternative. The reason that we've never really heard about it is that in the year 1906, in the spring, he caught pneumonia and died, leaving his alternative vision for the science of inheritance unfinished and unpublished. And the Mendelians, one could say, won by default. So what is it that Weldon wanted for the science of inheritance? Well, a wonderful window into Weldon's way of looking at it all can be recovered from a document that lies in the archive at University College London. And it was very exciting for me as the historian of all this when I realized what I had, because this document it shows has the notes that Weldon used in presenting on the topic of inheritance to a general audience at the British Association for the Advancement of Science meeting that took place in Belfast in the autumn of 1902. So we know, right, that Weldon doesn't think that you should start with Mendel's peas and then complicate everything. So where do you start in Weldon's view? Well, Weldon that evening started his audience off with the organism you see here. Some of you might recognize it, some of you with biological backgrounds. It's a water flea, or in its Latin name, Daphnia. Weldon told his audience about experiments done when he was back at UCL in the late 1890s, where Daphnia were kept in a beaker, and as the water that the Daphnia were kept in got polluted with their wastes, the spines of the Daphnia began to shrink until eventually spineless Daphnia were born. But Weldon went on to tell his audience, if you remove the spineless individuals to pure water, the descendants of those individuals will grow up to have a full, long spine. So here is how Weldon spelled out the take-home for his audience. Remember, the title is Inheritance. Now, clearly the condition of the spine, says Weldon, is not exclusively acquired, right? It's not exclusively from the environment, and it is not exclusively inherited. It belongs to both categories. A Daphnia of certain ancestry, right? We now say genetic background, growing in a solution of certain chemical composition, has a spine of a certain length. You cannot assert, says Weldon in 1902, that either factor, the inherited or the acquired, is more essential than the other. For if the Daphnia remain the same, you can change the result by changing the solution, while if the solution remained the same, you can, within considerable limits, change the result by changing the Daphnia. We see here, says Weldon, the complex way in which the characters of an organism are determined by the interaction of the two sets of factors, the environmental and the ancestral. In Weldon's view, that's where you start. If you're getting serious about inheritance in the early 20th century, that's the great generalization. That's the beginning point. If there's anything that's generally true about inherited characters, it's this point about interaction and the complexity of it. Now, Weldon, as I said, died in 1906. So far as we know, he never gave this point graphical expression. But in 1909, a German biologist called Volterek, also working with Daphnia, did exactly that. And what you're looking at here are the first norm of reaction curves, to use Volterek's enduring vocabulary. What you see on the y-axis, the vertical axis, is the value of the particular character, to talk jargon talk of geneticists, the phenotype. Pardon me. Here, not spine length, but head length. And on the x-axis, the horizontal axis, we see the environmental variable. Here, not water quality, but quantity of nutrient. And each curve represents a different variety of Daphnia. So what you see at a glance with a norm of reaction curve is that the character, the visible character, right, the phenotype, is the variable upshot of particular hereditary constitutions, particular genotypes, in particular environments. Both matter. And that's a point which is very easily missed with the typical way of representing a basic Mendelian experiment, the Punnett square. Reginald Punnett being one of William Bateson's lieutenants there among the Cambridge Mendelians, right? It's never really drawn to our attention that a Punnett square is exclusively to do with genetic variants, alleles in the jargon. You could easily miss the fact that environments matter too by looking at a Punnett square. Now, uh, from 1902, Weldon began to develop his positive program for the science of inheritance. And as I see it, you can summarize it in terms of three dicta and an upshot. The first is that variation matters. In Weldon's view, we should want a science of inheritance which is true to the variation that we actually observe in the world, not a fictionalized, idealized, cleaned up version of it. That's what we want to explain, what we actually observe. And we mustn't forget that. So, first, variation matters. Secondly, ancestry matters. What does he mean by this? What he means is that we don't just inherit a factor for this and a factor from that from our parents. We inherit a whole suite of factors, what we now call genetic background. And it's never really drawn to our attention, but when we study, when we examine an elementary Mendelian pattern, we're never invited to be curious about the parental organisms at the start. We're told that they're true breeding, and our curiosity shuts down at that point. Weldon thought that was a mistake. Weldon thought that there could be patterns which only disclose themselves if you attend to the longer run of a particular family lineage, because the particular lineage makes for a particular ancestry, particular background, and that can matter to the visible effects of any bit of chromosome. Finally, environments matter. And Weldon thought of what was going on inside a body as an environment for a chromosome, as well as what was going on outside a body, the physicochemical environment. Right? A chromosome doesn't just have its effect on a body in a kind of vacuum. It has its effect on a body within the context of lots of other things happening physiologically and physicochemically. And all of that can matter to the visible effect, right? All of that can impinge upon the chain of causation from chromosome to what we observe. So the upshot of this, in Weldon's view, was that the Mendelian picture of dominance as absolute, right? Where if the P has a yellow making factor, the P is going to be yellow, and you don't need to ask about anything else, right? Yellowness is dominant to greenness, full stop across the board, no matter the context, was at best deeply misleading because context always matters in Weldon's view. And when it doesn't matter, when it doesn't seem to matter, our curiosity should be excited. When it doesn't seem to matter to the point where you can get those pretty Punnett squares, it's because all of the variability that ordinarily obtains within an organism and without an organism is absent for some reason or other, whether for natural reasons or often for human engineers ones, right? It's not drawn to our attention how often the textbook cases of Mendelian inheritance are domesticated organisms, ones where we've engineered out all of that variability, where we've controlled for the environments. And uh, by way of helping you into the Weldonian perspective on Mendelian experimentation, I want to share an old joke. This is bound to be familiar to some of you, but maybe there's some out there that haven't heard this, right? So joke goes, it's nighttime. Guy's walking down the street, sees another guy down on the ground looking around. First guy says, "What are you doing?" Second guy says, "Well, I dropped my keys on the other side of the street." The first guy says, "Well, why are you looking for them here?" The second guy says, "Because the light's better here." Oh, good, I see a smile. So, on a Weldonian view, a Mendelian experiment is looking where the light is good, not necessarily where the key lies. And that's a very general preference in science, right? For looking where the methods that we're comfortable with, where we can apply our skills, where those methods enable us to go. And you can convince yourself that actually what you were looking for after all is what those methods disclose. But Weldon's here to remind us, not when it comes to inheritance. Now, the Mendelian perspective that Bateson championed, and without serious opposition, the most serious opposition, in my view, after Weldon's death in 1906, of course, is one of the great success stories in 20th century into 21st century science. Within living memory at my own university, the University of Leeds, a textbook was used for teaching basic genetics, iGenetics, and it came in two varieties, a Mendelian approach and a molecular approach. But they were the same chapters in different orders. In the molecular approach book, as soon as you got to whole organisms, as opposed to DNA, RNA, proteins, and so forth, once you hit visible organ or whole organisms and their visible traits, boom, you're right back again to Mendel and his piece. It's had a privileged place in the basic curriculum in genetics for over a century. And yet we know how unrepresentative it is. Even if you go, as I hope you will, if you haven't been yet, to the Mendel Museum in Brno, as you're heading towards the exit, having been through a beautiful exhibition all about Mendel and about Mendelian inheritance, you get a final panel which says, oh, by the way, things are actually more complicated than we've let on. It's like the fine print at the bottom of an insurance policy. So here we now get to the fine print just as you're leaving. They say, oh, by the way, uh, polygenic inheritance matters, right? Polygenic inheritance, as we see here, means that a given phenotype is coded by multiple genes. This is the case for the vast majority of traits, which makes their heredity a very complicated matter, right? The general case is not like the simple yellowness, greenness case. And that's true because usually there are many genes involved, not just the one. Secondly, it's because there are environmental effects. Here's how the panel puts it. You've no doubt noticed that if you don't water a plant with genes for tall height, those genes won't be enough to make it tall. The environment has a powerful effect on how our genes are expressed and produces even greater variability throughout populations. So no one's in denial. No one who teaches genetics is in denial about all of this complexity. And yet the traditional start with Mendel approach has remained static from Bateson's day to our own until recently. And I'm delighted to share with you my own role in beginning to teach genetics differently. And I came to have a role in this story as a historian of science, looking at the debate between Weldon and Bateson and asking myself, could it have gone differently? What if Weldon had lived that bit longer? What if he had lived long enough to publish the book manuscript that he was working on and it had had some purchase on the world? What would genetics be like now? What would genetics students be like now? And of course, thinking further, I realized that I couldn't actually go back in time, plainly, and give Weldon a bit more life to see what would happen. But it occurred to me that I could potentially teach a course in introductory genetics, which was as if it came from the Weldonian past that never was. So in this Imagine course, I start the students off not with Mendel's P's and then complicate things, but something which is genuinely representative of the role that genes play in producing the kinds of characters that might matter for them. Let's say the condition of a human heart. Genes are going to be part of a multifactorial causal mix, and a fascinatingly complex one. A mix that's going to change over the course of a lifetime of an individual. And of course, it's going to matter whether this individual is male or female, what social class they're in, what racial or ethnic group they're a part of, where they live, what they do, what levels of stress they experience, which again itself is related to many of those other factors, and so on. A multifactorial story where genes are absolutely in the mix, but one can't think about the causes that produce the condition of a human heart and conclude that there's a gene for cardiovascular disease, right? When one realizes what a simplification it is to speak that way when one's thinking properly multifactorially. So let's suppose in this Imagine class we could imprint the students, anchor their understanding on characters like this, and then throughout the course, hammer home interaction. genes with genetic background, genes with wider environment, the whole package, and all of the variability it produces, not as a kind of cognitive luxury item that the students can learn and forget, so that all they remember is dominance, recessiveness, right, and so forth. But instead, it's what the course is about. That's what genetics is about. It's about that interaction and the complexity it produces, so that when they do meet Mendelian patterns, they meet them as a special case. It's not that they don't matter, but they're a special case and they're exciting precisely because they're unusual. Suppose I could teach such a class. What would the students be like? In particular, might they be less prone to a kind of determinism about genes, right? The mistaken view that heredity is destiny. Might they be less prone to that error than students who are raised in standard, typical start with Mendel's P's fashion? Well, to my permanent amazement, I got a grant to run the experiment. I hired a brilliant postdoc, Annie Jameson, who had been a developmental biologist, became a historian of science, and so was really tuned in to Weldon's perspective. Uh, we collaborated on writing the curriculum. She recruited the students. She taught them our new Weldonian curriculum, this complexity first curriculum. And we assessed our students before and after teaching on their attitudes to genetic determinism. We did the same with students doing the standard introductory genetics course at Leeds. And we found something fascinating. What we found was that students who took the standard genetics course were, at the end of teaching, on average, as deterministic about genes as they were at the start of teaching. In other words, nothing they had learned in the interim had disabused them of the notion that heredity is destiny. By contrast, students taking our experimental Weldonian complexity first course, at the end of teaching, on average, were less deterministic in their attitudes to genes than they were at the start. And at least among the genetics educators that I spend time with, that's a win. The more you learn about genetics, the less confident you should be that you can just read off phenotype from genotype, read off visible character from hereditary constitution. We published the work first informally in Nature in 2016, and then formally in the journal Science and Education in 2017 under the title Genetic Determinism in the Genetics Curriculum, an exploratory study of the effects of Mendelian and Weldonian emphases. And I'm delighted to report that since then I've been collaborating with American colleagues on a next level of rigor, 2.0 version of the study, where in addition, we've been trying, and it's quite difficult to do this, but we've been trying to get at hypothesized links between categorical thinking, right? Thinking in terms of yellowness, greenness, or black and white, or having Tay-Sachs disease, not having Tay-Sachs disease, and stereotyping, whether that's in terms of sex or race or anything else, and the difference between what some psychologists call a fixed mindset and a growth mindset. A fixed mindset being a mindset where one thinks, I can't change, the world can't change, things just are the way they are. The growth mindset being the opposite of that. And this can matter, right? This can matter for thinking about not just peas, but thinking about people. It was left to me to draw the attention of one of the readers of my book, a very distinguished geneticist, who wrote to me to say, I'm really sorry to break it to you, but you've made a mistake on page 1 of your book because you at least imply that someone could have the gene for cystic fibrosis and not have cystic fibrosis. But we know, said my correspondent, that the cystic fibrosis gene, so-called, is 100% penetrant, meaning if you've got it, then you're going to have cystic fibrosis. So it was left to me, the historian of science, to draw the attention of this correspondent to a paper published in Science in 2015. It was the first study done where an attempt was made to examine the genomes of healthy people. So not starting with the genomes of people who are afflicted in some way and trying to figure out what is it in their genomes which is causing this affliction. Starting with the genomes of healthy people and asking, might they be harboring genetic variants which, according to our textbooks, should lead them to be dead, or if not dead, severely afflicted, and yet they're healthy. And if we can find those people, might there be things we can learn about what's going on in their genomes, which might enable us to develop new kinds of drugs? So the first time anybody attempted this, they found those people. And within the sample that they looked at, they found three individuals who should have had cystic fibrosis but didn't. It's something that matters historically when we look at the distressing history of connections between the science of genetics and eugenics, right? The program to breed better people through science. And in my book, I write quite a lot about the connection between elementary Mendelism and the eugenic program, because elementary Mendelism lends itself to eugenic purposes in leading leading to the thought that if you want to get rid of the problem, you get rid of the gene. And you get rid of the gene by getting rid of the people who harbor the gene. And as I point out, the two places where public Mendelism became most enthusiastically embraced were also the two places where eugenics went to its most distressing extremes. Those two places being the United States in the 1920s and, of course, Nazi Germany in the 1930s and 1940s. Now, you might say that's all well and good, but when we think about teaching genetics nowadays, isn't it best, nevertheless, to keep things simple? Sure, there might be all sorts of problems with starting with Mendel's peas. It encourages a determinism that our biology no longer acknowledges. It encourages an essentialism about people, which seems socially problematic. But what else are we going to do? Don't you need to keep things simple? Isn't that just timeless pedagogic wisdom? Well, I think one interesting answer here, but we could maybe talk more about it in the Q&A, was from the psychologist Stephen Heine in his book DNA is Not Destiny, published in 2017. Actually, at this point in the book, he's commenting on the experiments done at Leeds in genetics pedagogy. But here's what Heine had to say about that. There's a key difference between the complexities of the atom and the complexities of our genomes. We don't make life decisions on the basis of our understanding about how subatomic particles operate. Right? Incorrectly believing that the atom is akin to a miniature solar system has no consequences in our daily lives. In stark contrast, incorrectly believing that genes are like switches leads us to become fatalistic and can result in increased sexism, racism, and irrational fears about our future disease risk. When it comes to our genomes, I submit that it's far better to highlight the difficulty in understanding the complex machinery of genetics than it is to give people a false sense of understanding that leads them to make potentially costly decisions in their lives. So, to draw to a close, what I've tried to do in busting some myths about Gregor Mendel was first to emphasize that the notion of him as the discoverer of the gene, conceived of as the atom of heredity, has no basis in the historical record. And indeed, when we try our best to leave the present behind in reading Mendel, what we find is someone who was interested in the first instance not in heredity, but in hybrids. And when he conceived of the material, the underlying cellular material that explained the patterns that he found in his hybrids, it's at least plausible that he conceived of them not in terms of little atoms, but in terms of fluids, like immiscible fluids. Secondly, that as we've seen, the notion that an enlightened understanding of how biological inheritance works traces back to Mendel's garden uh, isn't a reflection of historical reality. It's a bit of polemical propaganda, uh, which the Batesonians, right, the Mendelians based in Bates and Cambridge, successfully put into circulation, and it's reflected in the textbooks that we all use. It's what we grow up with. But there was another way of looking at it. There was a serious pushback at the time, and indeed in that place, right, in England in the early 20th century. And the reason that we learn about Mendelian patterns first, when we learn about inheritance, rather than patterns in which the role of the environment in making for variability is conspicuous, is not because of how nature is, it's because of debate that most people have never heard of. The debate between Bateson and Weldon ended the way that it did. And in my view, it could have ended differently. And in learning about that alternative that might have been passed, we have a way of gaining critical distance on the legacy that we've inherited, on how we teach genetics. And out of that, I'm really pleased to say, has come a new way of teaching genetics, which is becoming more popular as people find out about it and learn that indeed there is another option and potentially a better option. I want to conclude with a few final thoughts about Gregor Mendel. Nothing that I've said, I hope, has suggested to you that I think that Mendel himself is in any way to blame for the conceptual errors, for the social difficulties which have followed from certain forms of Mendelian practice. As we've seen, Mendel was no Mendelian. And I think that if Mendel is to be taught about in schools, he should be taught about historically. So he shouldn't be taught just to fill students with slack-jawed wonder at the genius of Gregor Mendel. For sure, he was brilliant, and that brilliance should be appreciated. But the way to appreciate it is to put him back in his own context, to understand what his questions were and how outstanding the work that he did was in the service of those now hard to recover questions. And in my view, students who are taught to think about Mendel in this way have at least the ghost of a chance to bring that notion that even the best science is always part and parcel of its times. They can bring that knowledge to themselves to become reflective about how their own scientific work, if they go on to become researchers, or if they don't become researchers, how the scientific work which they have to make their minds up about when it's time to take decisions about their own health. Uh, all of that is part of a social world because there's no other way for science to take place. So to take that knowledge from a study of Mendel and bring it to the present seems to me an entirely salutary way to let Mendel into our lives. And I'll conclude there. Thank you very much.
Thank you very much, Professor Radick. Just for sharing. Just to start to kick off questions someone sent me. If you said Mendel intrinsically wasn't a geneticist and only his focus was on hybridization, how did he get the reputation of being the father of genetics? Would he, don't know if you can answer, would he want that reputation? I mean, not that you obviously know his feelings.
Well, that's a great question. Thanks for all the claps. It's very encouraging, having held forth for a while. Um, uh, the uh, the reason that Mendel got that reputation is that biology changed between the period when he conceived of his experiments and then lectured on them and published them. So between the mid 1850, around about 1865, and the later part of the 19th century, the biological discussion changed. And one way to think about it is that whereas for Mendel, hybridization is an end unto itself, right? He really wants to understand hybrids. That's an interesting question. These two classes of hybrids, the constant and the variable, and the laws that govern them. and the explanations of those laws. For Mendel, that's an end unto itself. By contrast, by the end of the century, hybridization had become an end to the means of understanding, had become, I beg your pardon, had become a means to the end of understanding something else, something grander, heredity and evolution. And so the new way of thinking about crossing experiments as throwing light on these larger questions is read back into Mendel's work. So he's retrospectively assimilated to the later conversation. And that's why he's read as having contributed in the first instance to this debate on heredity and evolution. Would he have been pleased? That's a really interesting question, and not one that I thought about. Mendel, though we tend to think about him as humble, right? Because we think about him in his humble garb there, as we saw in the photograph. Mendel thought that he mattered. Mendel thought that he was going to be important scientifically. And as I said, by becoming abbot, his scientific career ended sooner than he expected that it would. But Mendel thought he was going to be famous. So I think the idea of being lionized around the world as one of the greats of all time in science, I think would have appealed to Mendel very much indeed. The fact that it was for something that wasn't quite what he was aiming at, probably he would have been relaxed about that. But certainly he didn't want to hide his light under a bushel, to be biblical.
Okay, couple of questions in the chat. Is there any suggestion that Mendel's segregation data was too perfect and he may have fudged some of his results?
Yes, excellent. And indeed, the first person to point out that statistically Mendel's results are improbably close to his theoretical expectations was Weldon. And indeed, it was in that very paper that I showed you the P photograph from that 1902 paper. So Weldon points this out, but for Weldon, it's not a hanging offense. Uh, for him, it's a symptom of the underlying problem. The underlying problem being Mendel's use of the binary categories, right? If you've persuaded yourself that binary categories are appropriate, then what are you going to do when you're confronted with a P, which is not either yellow or green? Well, you're probably going to put it in the category which your theory leads you to expect needs the bump, the one or the other. But in Weldon's view, what was really important about the statistical too good to be true fact, as he saw it, was what it told us about categorization, about Mendel's descriptive categories, and the way that those descriptive categories end up leading Mendel to deal with the hereditary constitutions in ways that suggest that if you've got one hereditary constitution, you get this blocky binary character. So that's the mistake. I've actually written a paper called Mendel the Fraud, a social history of truth in genetics, which traces the history of the debate. Because as I say, Weldon didn't think it was a hanging offense. And for decades, geneticists didn't really worry about it. They talked about it, but they didn't worry about it. It became the kind of thing that everybody else now knows about, so that it gets raised in a forum like this from the 1960s and 1970s. And what I try to suggest in that paper is that that's for interesting reasons to do with changes in the Cold War and the relationship between the Cold War and the sciences. But I'll leave that to you to have a look at in the paper if that's of interest.
Thank you. And last last few questions in the chat, then we'll wrap up, I think. Are you suggesting that the current technique of gene manipulation, sorry, let's just move on to something, that go towards curing diseases are leading us down the garden path.
No, not at all. Well, uh, and I mean, this is the week in which really exciting gene therapy results were announced for combating Huntington's disease. And so I don't in any way see myself as suggesting that we don't have we don't rightly expect lots of good things to come from genetic medicine. That said, we have to beware myopia about it, and it's distressingly easy to overfocus on successes in combating what are really quite rare diseases and not to pay enough attention to environmental factors that affect all of us. And I think one of the most exciting areas of research, at least to my mind, is drawing out the extent to which air quality, for example, figures in complex ways in a whole suite of diseases. And so we shouldn't be skeptical about the promise of genetic medicine, but we need to keep that in perspective. So it's not leading us up the garden path, but it's up to us not to forget all of the ways in which environments matter, right? As I emphasized in the talk, internal environments and external environments. So we should be we should be pleased and eager to learn more about what genetic medicine has to offer. But we should keep that in perspective and bearing in mind the role of internal and external environments in making for variability, I think is an important an important guard against expecting too much and then being disappointed.
Thank you very, very much, Professor Radick, for a really, really fascinating talk. Different to our usual lectures, but definitely equally, if not more interesting about how the whole field came to be. And I certainly learnt a lot. Hopefully, we'll have our next lecture in around early to mid December time. Hopefully, I'll have more details in the coming weeks about that to send out. And if you have any other questions, I'll pass them on to Professor Radick and see if the answers can liaise between them. And hope everyone has a lovely evening and a Shana Tova.
Thank you, everyone, for your attention. It's been a pleasure to be with you this evening. And thank you to Gidon and to Josh for hosting.